Sea lice, also known as L. salmonis, are known to frequently parasitize farmed salmon through frontal filaments that attach to the skin of the fish. To assess if salmon have molecular defenses against host recognition molecules, researchers have sought further understanding of the naturally occurring deployments of antimicrobial peptides, or AMPs, possessed by salmon such as cathelicidins. Cathelicidin 2 has been identified as a mediator of early innate immune responses and has been seen to increase in expression when challenged by infection. Ergo, they are highly sought after for the experimentation and require methods to significantly increase in the quantity. Leishmania are unicellular protozoan parasites that possess a well-defined nucleus amidst a variety of organelles and a flagellum that provides them with their motility. In their amastigote forms, they appear as globule-shaped cells, often appearing in clusters and replicating patterns before transitioning to their promastigote form, appearing as an elongated teardrop shaped with a singular flagellum. Leishmania tarentolae have been investigated for their intrinsic plasticity of their genome and suitability to produce complex recombinant proteins and peptides that contain post-translational modification capabilities, something that prokaryotic systems cannot do. Rich in glycoproteins, Leishmania can create folds and linkages within complex proteins, such as cathelicidin 2. Upon discovery of 2 stocks of cryopreserved Leishmania tarentolae that, due to their labeling patterns, were presumed to be transformed and non-transformed from a previous study, it was hypothesized that Leishmania tarentolae can be genetically transformed with a cath-L2 containing expression cassette to propose an expression system capable of expressing cathelicidin 2 through investigative confirmation. At least one of these recovered stocks were thought to contain recombinant DNA from the plasmid vector, Lexisat 2.1, and expression cassette region containing cath-L2, a gene that expresses cathelicidin 2. Transformational and clonal capabilities of E. coli were also investigated through a process known as electroporation, whereby a single high-voltage pulse was released into a sample containing E. coli, and the Lexisat 2.1 vector, creating pores within the membrane for transformation, was selected through ampicillin plating before an individual colony was grown overnight in preparation for plasmid extraction. The 2 recovered Leishmania stocks, as well as the kit-provided strain known as P10, was cultured within supplemented RPMI media for 5 days, and observations on the motility, confluency, and overall health were made throughout the growth process. Selection of recombinant Leishmania was performed with norsothricin antibiotics, to which successfully recombinant strains would be resistant. After the selection period of Leishmania and the growth of recombinant E. coli, plasmid DNA was extracted for diagnostic PCR analysis of the amplicons of 2 regions along the expression cassette. The upstream flanking 5' small subunit rRNA to the untranslated region number 1, and the insert sequence, to which the cath-L2 sequence was thought to be spliced into previously. It was predicted that the only amplicon that would be present from the 5' SSU to UTR1 flanking sequence would be the transformed Leishmania sample, as one primer requires a sequence belonging to the Leishmania DNA outside of the cassette, while the reverse primer begins within the cassette itself. The insert sequence was predicted to amplify only within the Lexi-neat sample, transformed E. coli, and the transformed Leishmania, as they were the only samples thought to contain the vector and/or expression cassette. Qualitative PCR results had determined the trend of amplification throughout all of the 5' SSU to UTR1 control region samples from the non-transformed E. coli, transformed E. coli, Leishmania P10, non-transformed, and transformed strains. Agarose gel electrophoresis confirmed the size of these amplicons that did not match the intended base pair length of 1,635 base pairs. Conversely, the only amplicon that arose from the priming of the insert sequence of the expression cassette was the transformed E. coli. Therefore, confirmation of recombinant LexCSAT 2.1 E. coli was confirmed through qualitative PCR and gel electrophoresis. However, the lack of growth of recovered Leishmania tarentolae stocks in culture media, as well as DNA extraction oversights, did not allow for confirmation of the integration of a cathL2 containing expression cassette into Leishmania tarentolae. Future research into this topic should include increasing the transformation efficiency of the Leishmania vector into E. coli by decreasing the concentration, replacing rPMI media with BHI media, acquiring restriction enzymes for restriction digestion analysis, avoiding antibiotic selection processes greater than 3 days, and in place of plasmid extraction for Leishmania tarentolae stocks, extracting mRNA from the cultures, as Leishmania do not hold plasmids in their native form. Converting mRNA to cDNA for PCR amplification must happen. I would like to express my sincere gratitude and appreciation towards the Holland College Faculty of Applied Sciences, namely Dr. Michael Gibson, Dr. Jessica Livingston, Dr. Jen Slemmer, Crystal McFadyen, and Marla MacKenzie for their guidance and support of this project.